The second part of this module is going to focus on one of the functions, which is the regulation of substances in and out of the cell. So the plasma membranes are selectively permeable. Okay, what does this mean? They allow certain things to pass through, but not others. Okay, so think about that. The cell membrane has to kind of prevent from harmful materials from entering the cell, right? So they don't want those things to go into the cell, but they also want to leave things like nutrients in the cell, right? They don't want them to leave the cell. So it can be very, very selective in what it allows to pass through. So the cell membrane usually allows lipid-soluble materials to pass through. And why is that? Because that fatty acid tails, right, which are nonpolar. So the nonpolar materials, which is lipophilic, they're like lipids, they're lipid soluble, those materials can pass through this fatty acid tail section very, very easily. Okay, so usually they don't have any trouble passing through the cell membrane. But there's a little catch there. They can't be too big. They have to be, relatively small molecules. So things like fat-soluble vitamins, vitamin D, vitamin E, or some hormones like steroid-based hormones, like sex hormones, those molecules can pass through the cell membrane very easily. So you can say that they can cross the cell membrane freely. Other examples include some gas molecules like oxygen, carbon dioxide. They don't carry any charge, so they can also, move through the cell membrane very easily. And you actually want that, right? When you have oxygen outside the cell, you want the oxygen molecules to, diffuse very fast into the cell so that your cell can utilize the oxygen to generate energy. And the waste product, the carbon dioxide, you want it to move out of the cell very quickly. So you want those two gases to cross the cell membrane very easily. These substances, on the other hand, are going to be kind of stopped or blocked by the fatty acid tails. And usually these substances are polar molecules. So they don't like to go through this fatty acid tail section. Okay, so they can't cross the cell membrane very easily. Examples are ions, because ions are charged, so they're polar. They don't like fatty acid. Simple sugars, amino acids, they're also very water soluble. They don't like the fatty acid, that section. Okay, so they can't usually cross the cell membrane freely. Okay, so now all those different things, they can either cross the cell membrane very easily or they can't. But a lot of times we need to transport these things into the cell membrane, right? So that determines that we are going to have a different mechanisms of transport, right, to move things across the cell membrane. So we're going to have three main types of transport. We're going to focus on these three. Passive transport, active transport. So these are going to transport smaller molecules. And if you need to move something really big across the cell membrane, you have to resort to the last one, the bulk transport. Okay? The main difference between passive and active transport is that whether energy is required. Okay, so passive transport does not require energy. Things can just move, but active transport requires energy. So you have to provide energy for things to move around. All right. So we're going to focus on passive transport first. Like I said, it does not require energy. Molecules can just move from a higher concentration to a lower concentration. Okay? So think of this. You probably have all been on a slide when you were a little kid. So when you slide down, when you move from the top to the bottom, you're going from a high ground to low ground, and because of gravity you do not have to do any work, right? You just slide down. So that's not perfect, but a pretty good analogy for passive transport. Okay? So things move from higher concentration area to a lower concentration area, and because of the kinetic energy in those molecules, that energy kind of make the substance move or with a tendency to move from high to low concentration. So there's no energy required. Okay? Now in passive transport, we have a few kind of subcategories, different types. The first one is simple diffusion. So things can move freely from high to low concentration areas, and there's no carrier proteins, no help at all. Okay? So the molecules are on their own. Facilitated transport or facilitated diffusion. So facilitated, that means somebody is helping those molecules move. And the little helper is actually protein. Okay? So they could be protein channels or they could be protein carriers. Okay? Here are channel proteins. The last type is called osmosis. So this is really about the passive transport of water. Okay, so that does not require any carrier proteins. Okay, so this is a special. Oops. So osmosis is really a special type of simple diffusion because it's really just a diffusion of water. Okay, so let's look at a couple examples. So we say in passive transport, molecules move from higher concentration to lower concentration. So this is what happens. Okay, so let's say this is the plasma membrane. This is extracellular space outside the cell. This is the interior of the cell, intracellular. Okay, so if you have a concentration gradient, so what is concentration gradient? Different concentrations. So in the extracellular space, you see these are specific molecules. The concentration is really high, right? But in the intracellular space, inside the cell, there's no such molecule. So this creates a concentration gradient because the concentrations are different across the cell membrane. So over time, if this molecule can cross the cell membrane easily or freely, then over time, then you can see that molecules tend to move from this high concentration to low concentration area, right? So it's going to move to the interior. And if you give it enough time, eventually it will reach an equilibrium. Equilibrium just means the concentrations across the two sides, they're going to be the same. All right. So that's a simple diffusion of this particular molecule. Okay, so there's more detail over here. Now, a lot of times, simple diffusion just happens in our daily life. For example, sugar in coffee, when you add sugar, you kind of stir it a little bit, right? So what you're doing there is you're kind of accelerating or speeding up the simple diffusion of sugar to make the sugar distribute evenly in the coffee, right? To eventually reach that equilibrium state. Aerosol spray, so if you spray something, the molecules are going to move from the high concentration area where you just sprayed to lower concentration. That's the ambient air, other space. So people that may be a few feet away from you can also smell the molecules, right? So that's the diffusion of those aerosol molecules. Okay. Same thing for someone's perfume, right? You can smell someone's perfume because the molecules diffuse from the person who uses it, the high concentration, to other areas with the low concentration. And then when you walk past by, your nose can pick up those molecules, and this is how you can smell someone's perfume. So all those things can diffuse very easily through the cell membrane. So those are the things that we talk about, right? The oxygen, the different gases, and lipid-soluble molecules. Okay. Now, factors affect the rate of diffusion, extent of the concentration gradient. So the higher the concentration gradient is, meaning the difference is greater in those two areas, that's going to increase the rate of diffusion. Okay, so let me give you an example. All right, so I'm going to make up some numbers. So let's say on one side of the cell membrane you have 10 milligram per liter concentration, and then on the other side it's 5 milligram per liter. Okay, so there's a concentration gradient, right? Because there's a difference between those two. But if we can make the concentration gradient greater, So instead of 5, now I'm going to change the concentration to 1 gram per liter. So the difference here is 5, right? But the difference for the second scenario is 9. So this is a greater concentration gradient. So the diffusion will be faster. Okay? So the rate, diffusion rate, will be higher, okay, than the first scenario. So that's what it means. Mass of molecules diffusing. So this just means smaller molecules will diffuse faster. And you can understand that, right? Smaller things can go through small space easier, so they can move faster. And larger molecules will move slower. All right. And the temperature. So higher temperature provides energy, provides more energy, right? Provides more energy. And with that extra energy, molecules can move faster, right? So diffusion, so it will be higher rate of diffusion. Solvent density, you can skip this one. This is not very common one. Solubility, so that depends on the lipid solubility, right? If you're more lipid soluble, then you probably will diffuse faster. Surface area, thickness, okay. So more surface area, more diffusion. The thicker the plasma membrane, the less easier or more difficult for things to move through. So greater surface area will increase diffusion rate, but thicker plasma membrane will decrease diffusion rate. Okay? Distance traveled, diffusion. Shorter distance, faster diffusion. And then longer distance, slower diffusion. Okay? So longer distance, slower diffusion. And then shorter is the opposite, right? Faster. Okay. Now the second type of transport is facilitated transport. So ions, polar molecules like glucose, they are really repelled by the hydrophobic fatty acid tails. So they need a little bit help. Okay, so we are going to have proteins in the cell membrane to provide a special corridor. Okay, now MBN just stands for membrane. So these proteins are going to help those molecules to move across the cell membrane. Okay, so this is the mechanism. So they shield these molecules from the repulsive force of the fatty acids. So we have two types of helper proteins. The first one is called channel proteins. They will form channels, and a lot of times these channels will help ions to move through. We may also have carrier proteins. So for example, glucose transporter. So this particular protein can carry glucose and move them from outside to inside of the cell. Okay, so this is an example of a channel protein. So you can think of these green things as ions. Okay, so we need to move these ions from the exterior to the interior of the cell. But these ions are polar, so they're really repelled by the cell membrane. So even though there's a concentration gradient, they can't really move. So what happens is you're going to provide this protein channel. And when the channel is open, because it shields, right, these molecules from the fatty acid tails, so that these ions can just move by the concentration gradient, and they're going to move through the protein channel and then into the interior of the cell. Okay, so this is how it works. This is a carrier protein. So it's very, very similar. They can bind to a substance, and then this will trigger change of its own shape. And when the shape changes, it will move the molecule, to the other side of the cell. Okay, so this is how it transports polar molecules. So this is very similar. There's a protein channel, specifically ion channel for potassium, and this is a carrier protein for glucose. Okay, so you can see the first step is glucose binds to the carrier protein, and when that happens, the protein carrier changes its configuration, right? Changes the shape. So it's going to open up on the other end. So this is how it can move the glucose across the cell membrane.